Coronavirus testing swabs, offered as a public service, brought to you by a biochemist approved by a medical doctor. Their names are kept confidential. Hila says, while studying bachelor's degree in biochemistry and food sciences, I studied microbiology. We grew bacteria from a certain food product in agar petri dishes that after a week or so grew colonies, yeast, and mold as well. Throughout the years while I worked in food factories, we always checked different kinds of pathogenic bacteria that could cause illness such as staph aureus, E. coli, yeast, and molds. We never checked for a virus. There are some tests for viruses in food in Europe, but in Israel it was agreed that there is no specification to mandate such tests. I think that the COVID tests are not reliable because while bacteria such as Streptococcus that could cause a throat infection will grow in colonies on a petri dish, that is if one was infected by them, and if not, the doctor will say you have a virus. A virus, unlike bacteria, yeast, or molds, cannot grow independently without a host, animal, human being. That is why diagnosing someone with a virus is through negative result for bacteria, apart from blood test samples that results arrive a few days later. That is why I find coronavirus testing positive peculiar. In order to detect a specific virus, you need to do a thorough test under a microscope and use few methods to isolate it. This is an expensive and long procedure. The chances that swab tests taken from the nose or mouth test for presence of a virus or even a part of a virus are very small. And even if some of the virus found could be any virus, not specifically corona, because in fact it checks exosomes, and exosomes exist in your body and not only in viruses. I read that COVID test is a PCR test, so I asked two doctors do microbiology, and they confirmed that the swab test is a PCR test. Details on the test and the inventor of the PCR technique, Carrie B. Mullis, may rest in peace, follows right after this. It is possible to have serology tests taken from Maccabi site to detect presence of antibodies to different illnesses, bacteria, viruses, parasites in the blood, semen, and saliva. The test is used to diagnose different illnesses and certain medical conditions. Serology test helps determine whether there is an infection and also the stage of the infection, new infection or old. This is determined through the type and the amount of the antibodies found in the sample. Sometimes it is needed to test again after a few weeks to determine the stage of infection. 10 things you should know about the coronavirus swab testing for COVID. 1. The process used for COVID test has always been deemed an invention as it was never qualified as a test. This invention is called PCR, standing for polymerase chain reaction. 2. The inventor of PCR, Carrie B. Mullis, said that these PCR tests cannot detect infections-free viruses at all. The PCR was invented to merely have bits of genetic material in a sample multiply until those small broken pieces were numerous enough to see under a microscope. The microscope was developed by Anton van Leeuwenhoek about 300 years ago. I chose this as a research science project in elementary school. He was the first to observe bacteria and called them small animalcules. The secret of his special polishing of the magnifying glass he took with him to the grave, so another 100 years passed until his invention was recreated. The PCR just helps scientists observe small things in greater quantity. Where those bits of genetic material came from, what they do they make people sick, etc. The PCR process is not involved in that determination whatsoever. 4. In order to finger any virus as the cause of someone's illness, the entire virus, the complete genetic sequence, must be isolated. This has not yet been done with COVID-19. This is correct. No complete virus has not yet been isolated, and that's supposed to be the first step before any other steps are taken in such a process that ends with a viral declaration. let alone a pandemic declaration. 5. The entire virus must be transferred over to another animal or human host, and that virus must cause the exact same illness in the host. This has not yet been done with COVID-19. Why are these steps being bypassed? 6. In order to finger any virus as the cause of someone's illness, the entire virus must not only infect the host with the exact same disease, you must also find the complete genetic viral sequence in the infected tissue of the host, and then re-isolate the entire genetic sequence from infected tissues of the host. Disease transmission person to person has not been proven for COVID-19. This is the easiest criteria to organize, yet it remains undone. 7. In order to finger any virus as the cause of someone's illness, you must also have a control group, so you can make sure that what you call a virus isn't just a regular human genetic material shared by all humans. If you find the exact same 80% genetic material strand in the samples of healthy controls, what you're looking at can't be the cause of the disease, because it's just regular genetic material found in all humans, both sick and healthy. In order to finger any virus as the cause of someone's illness, you need to find that particular complete viral sequence in the sick only, and not in the healthy. Why is this gold standard of testing, having a control group, being bypassed? 8. In the case of COVID-19, scientists have indeed found a string of genetic material, not a complete sequence, that shares 80% similarities with SARS-CoV, a sequence said to cause SARS, and therefore the original Chinese scientists have declared that this string of material reflects a COVID infection, but also did that without testing any healthy people. Humans share 97% genetic material with chimpanzees, 80% genetic material with cats, and 50% genetic material with a banana. And that's why shared genetic material is never the standard used to diagnose any disease. To declare viral disease, you always need 100% viral genetic sequence. In the past, this so far has not been done for COVID-19. 9. The PCR invention shouldn't be used as a way to diagnose any disease because it's simply designed to make small bits of genetic material more visible under a microscope. Those small bits were always designed to be studied, not to conclude disease or no disease. That is why the PCR process has already been shown, in its current misused way, to declare 8 out of 10 people positive with COVID, when they're perfectly healthy. Out of 10 people in a COVID testing area, 8 will be told they have COVID. 2 will not, and there is absolutely no basis to any of those declarations whatsoever. So why are we using it? 10. You are more than welcome to ask microbiologists, medical doctors who study microbiology or infectious diseases, and scientists who studied HIV virus. You will be surprised. 11. People die, for sure, but this is not a pandemic. The swab tests, as you can see, are not reliable. It is possible that people died from different reasons, as it was probably from the beginning of mankind. Putting someone on artificial respiration devices when not in need will most probably lead a system failure and might lead to death. There is a respiratory aid protocol. Different methods, such as BPAP and CPAP, should be used beforehand. Putting a person with anxiety attack with 80 to 90% saturation of oxygen in the blood is not part of the protocol. These are quotes from different doctors. You are most welcome to investigate this yourself.